So we're going to kick off, starting off with the basic science of drug discovery. And we're delighted to have Dr. Finbarr O'Sullivan, who's the Associate Director of the National Institute for Cellular Biotechnology at Dublin City University. And Finbarr, you should be able to grab the talking stick there and share your screen.
Brilliant. Okay, so first I'd like to thank Midland Science for the invitation to speak this morning. Hold on, those quirky these things. Now, there we go. So, as you heard, I'm from the National Institute of Cellular Biotechnology in Dublin City University, and we've been gone to go in one guise or another since 1987. And our current sort of form and building, we've been in there since 2006 in DCU. And inside the NSCB, we've a number of different areas of research. We grow in vitro cell systems as research tools, and as therapy, and to investigate therapies and the likes for the likes of diabetes, coronary disease, to look at metallobiology, cancer, and we also look at how we grow cells and use them actually to produce pharmaceutical compounds in their own right. And as well as being part of the NICB, I'm also part of another institution, centre called SSPC, which is led from the University of Limerick. And SSPC actually it's a national collaboration with about 400 different members. And as you can see, it's not just UL, it's DCU, UCC, Athlone, Trinity, Waterford, UCD, and UL. And we work with a number of different pharma companies on sort of understanding how medicines work and how to improve medicines and how to improve their manufacture. And within sort of SSPC, there's a range of kind of, there's three sort of raw themes. There's one called molecules, where they're looking at the sort of how these molecules work and design them, materials where they kind of scale them up and how to make them, and then medicines in the formulation. And I'm sitting at the very start of that journey of where you're taking a molecule and you're trying to figure out, how does that actually work? Does it have an effect? And what are those effects? So, as I say, we create cell models and we use them to look at different things. We look at them to understand different diseases. We also look at how existing drugs work and how to improve their usefulness. And also, say, to look at, maybe reduction of therapeutics from cells themselves. And even now, I suppose, cells and working with cells and the way technology is going, we're now even looking at using cells themselves as a therapy or a medicine in their own right. So I mentioned what a cell is, and just to kind of clarify, I suppose, what cells are. So what is a cell? It's simply it's a simplest form of life. It's a structural unit that makes up everything. So even if you're a bacteria or you're a big multicellular animal or a plant, you're made of cells, and they're kind of the units of which things are made of. And I suppose the very first description where the name has come from, it came from Robert Hooke. He worked with Robert Boyle, and Newton's famous quote, which is, "If I stand on the shoulders of giants, it's because I see further." It was actually written in a letter to Hooke, but it was actually a bit of a dig at Hooke, because Hooke was actually a bit on the small side. So anyway, Hooke built a microscope, one of the very earliest kind of forms of microscopes, and he looked at different things like insects and feathers and rocks and pieces of cork. And it's the last thing that he looked at, cork, where he said, oh, they reminded him a bit of the sort of the wall structures that monks would live in. And he coined the term cell. And I suppose there are two broad classifications of cells. There's the bacteria, or given the fancy name, the prokaryotes. And that means the Greek, coming from the word Greek words before, pro, and nut, meaning kernel, and means that their genetic material isn't encapsulated. And they're quite small. We can just about see bacteria under a light microscope. The other ones then are the eukaryotes. And again, that comes from the Greek words eu meaning good and karyon again meaning nut. And really all that means is that their genetic material is encapsulated in a nucleus. And here we're talking about the animal cells, and obviously humans are part of what I mean animal cells, I include humans in that. Plant cells, fungi, single cell algae, they're all eukaryotes. And it's the animal cells and that I'm interested in. And that's what we work with in our lab. Now, as I say, they're a lot bigger than bacteria. They have lots of different things inside them that let them do different things. You don't really need to worry about that per se, but just know that they're far more complicated than bacteria. And that's what makes them more interesting to work with from a point of view of medicines, or difficult, how medicines will react with these things differently, and that. So just remember that cells in our body have different components, they do different jobs, and how they work. So when we grow cells in a lab, most of the time we're growing what's known as a cell line. And those cell lines as such are immortal. In other words, they'll keep growing indefinitely, as long as you give them the right kind of nutrients and food and space to grow in environment, they'll happily keep growing indefinitely. And the very first cell line was a cell line called HeLa, and it's still on the go today. And it was derived from a woman called Henrietta Lacks by George Gay. Henrietta Lacks was, she died very young. She had a very aggressive ovarian cancer. And the story of Henrietta Lacks and the story of HeLa, such, it was actually made into a book. Actually, it was a bestseller, and I think it became a film as well. And it's kind of a sad story. She was Black, so in America in the 1950s, there was only limited places that would actually treat someone who of African American descent. So she actually gave the sample that was used to create HeLa was actually something that she actually hadn't consented to. She herself died in extreme poverty, and I think even today her grave is actually not fully known. But anyway, that's something that's good to have a look at, is that book. It's quite good. So when we work in a lab, we work with cells. We work in a what we call tissue culture room, and we work in these kind of cabinets here called laminar flow cabinets. And they keep the air lovely and clean and sterile for us. And we grow our cells in these kind of flasks, as we call them. And the media there, that's the growth media that they grow. So the cells are kind of just a very thin, transparent layer on the bottom of the flask. And that media there, all that is actually salt, nutrients, and vitamins, and amino acids to help the cells, to give the cells the energy and building blocks to grow. The red color isn't blood, which a lot of people actually think. It's actually something that's a color just that shows if the media is becoming acidic. Because cells, as they grow, often produce acidic waste products. And it's an indicator for us that we might need to change the media color over for them. And we use then different specialized tools to look at our cells. We use microscopes to look at where the location of different proteins are. We use techniques to look at how what genes are turned on or turned off. And we also look at what proteins, and proteins are sort of the engines inside a cell. So genes will encode proteins as such, but proteins are really the business end. What proteins are turned on or turned off as well in various disease status and that. And that tells us how sometimes a drug might be interacting with a cell, how what targets might be there as well to discover new medicine. So they're the type of techniques that we're using to do that. And kind of just give you a very broad overview of something of how those sort of things are used. We have a research program looking at pancreatic cancer. Pancreatic cancer is, again, a particularly aggressive cancer in that it's detected quite late because there's no effective screening methods, and the symptoms usually occur quite late stage. And there's about 550 people diagnosed each year in Ireland with pancreatic cancer. And as I say, therapy options are limited, and hence, because it's quite late in diagnosis and limited therapeutic options, the survival rate isn't very good. So cancer cells in general can acquire resistance to chemotherapeutic drugs. And this is often why chemotherapy often can fail. So initially, your cancer cell can start responding to a chemotherapeutic drug and then stop. And the reason is that it's essentially adapted to the chemotherapeutic drug and is now producing new proteins to deal with that chemotherapeutic drug. But often it's not just the one that you started with. You find that's resistant to a number of different chemotherapeutic drugs. So we create cell line models in the lab to look at this drug resistance and see how does it work. And we look then at the gene and protein expression. And again, we see how different models of these respond to different drugs. So one of these is Folfirinox. It's a chemotherapeutic drug approach using pancreatic cancer. It's a combination of 3 different drugs. And postgrad who's working with me, Taylor Allen Coyle, she takes cell lines and she makes them resistant then to either one of those 3 different chemotherapeutic drugs or the combination. And what she does then, she looks then to see how do the cells, different pancreatic cancer cell lines that she has in the lab, that she's made resistant and respond to them, and how the different cell lines themselves respond. And that in turn gives indications of when we look then at the gene and protein expression, how does that work? And then can we adjust it? If we adjust those chemotherapeutic drugs, can we improve performance? So if you have created a model of something that's resistant to the combination drug, if you adjust that molecule, if you change the molecule around that chemotherapeutic, or you create a new analog, can you reverse the chemo resistance that those cells have? Another area that we work on then is on a thing called mRNAs. So these are kind of the newer side of things, and potentially maybe medicines in their own right in time. So mRNAs, they're small little RNA molecules, and essentially all they do is they regulate, they're another step in regulating what genes are made into proteins. So when we look at a disease like cancer, we see changes in those mRNAs, those small little RNA molecules. And they can mean, that in turn means that certain proteins will be either turned on or turned off based on the changes in those mRNA molecules. And here, Dr. Fiona O'Neill, she's creating cell line panels, and she's profiling these mRNAs, and then she's either increasing the expression of those mRNAs or she's decreasing the expression of those mRNAs. And she's looking then at how that will affect those different cancer cells. Will they improve response? Will it disimprove response? So mRNAs themselves potentially could be therapeutics, but it also may give you, again, leads into new targets for molecules then to be screened against, so that you might find, well, actually, this particular metabolic pathway is very important in making these cells resistant. And if we can knock this pathway out, we can improve the chemotherapeutic response. So can we find a molecule that will knock out one of those parts of that pathway? So it gives you leads into the screening process that you'd use for that. And I suppose finally say that, like, when we sort of reach sort of something, a conclusion in the laboratory, you go, great, we've found something, it looks really good. And you'll often hear reports of that in the media and everything else, that, oh, scientists in a paper published in such and such have found, a cure for something. In reality, there's an awful lot of steps from that very basic area where I am before we get into the lab. There's a whole regulatory framework, which I know Karl Cogan from the HPRA in the next talk will probably talk an awful lot more about. But we also have to do things like animal trials and things like that to fully prove that these actually work. Because again, a lot of things can work very well in a controlled lab environment, but as you get further and further into human therapeutics, it's harder to, so that there's more challenges there for these things to meet. So finally, just to say thank you. I mean, like all research, it's a group effort. So I'd like to say, there's a group of us that are busily involved in this. There's Professor Martin Kleins, there's Dr. Joanne Keenan, Dr. Fiona O'Neill, whose work I mentioned, Justine Miller, Donald Gorman, Taylor, again, whose work I also mentioned, Marta, Neve, and Andreas. And again, people who fund this work, because again, it's expensive stuff. So we're funded by the Pancreatic Cancer Research Fund in the UK, Enterprise Ireland, and then SSPC through SFI fund this. And we're very lucky as well that we have very good collaborations as well with a number of industrial companies like Alltech, Glantro, and Janssen. So thank you very much. I hope you enjoyed that.
Super. Thank you very much, Finbarr. That was great. It was you got through a multitude.
It wasn't too fast.
You really gave a very good feeling for, I suppose, the amount of work that goes into, almost for the general public, it almost happens behind the scenes to a large degree, and it's all the work that goes into actually forming the idea in the first place. I thought something really interesting that you mentioned a few times is the next, the next big thing. And of course, with the COVID vaccines, they're an example of a first in class.
Yeah, exactly. Yeah.
So look, we're going to hold the audience questions until we've had the three talks. So we'll move on to the second talk. But thank you very much, Finbarr, for that. It was super. So we're going to move on to our second talk with Dr. Karl Cogan, who is with the HPRA, the Health Products Regulatory Authority, which is the national competent authority for medicines in Ireland. So this is the organisation that really assesses the safety and the efficacy of medicines and decides whether a really good idea, as Finbarr has outlined, has demonstrated enough and accumulated enough evidence to become a real medicine that patients should be using. So you should be able to grab the screen there, Karl.
No worries, I'll just bring up my little laser pointer. So yeah, no, thanks. As I'm sure all your speakers over the last kind of couple of weeks is like, thanks very much for inviting me and getting a chance to speak here. I think it's a wonderful initiative. And then also congratulations to the organizers. Been some fantastic talks over the last kind of couple of weeks. So I've got some big shoes to fill, but hopefully I can do this area justice. So I'm, as Craig mentioned, I'm from the Health Products Regulatory Authority of Ireland, and my talk is really going to be kind of focusing on how does a medicine get a license. So how does it get a license to sell its product in Ireland? So Finbarr talked very nicely about like kind of drug discovery and so on. So how do we go from that discovery of a promising new compound to it ultimately getting a license that can then be sold in Ireland? So whereas Finbarr is very much at the coalface of the discovery, we're much more towards the end of the process where we kind of evaluate all of the evidence and then ultimately grant the license. But I'll talk about that a little bit more on that process in terms of how we do that now throughout the talk. But Craig very nicely introduced the Health Products Regulatory Authority of Ireland. So we essentially, in a nutshell, regulate health products. So we have this responsibility to ensure that different health products on the Irish market demonstrate their safety, their efficacy, and then also that they're produced to a really high standard. So there's regulatory documents that outline the very specific standards that different health products need to meet to demonstrate their safety, their effectiveness, and so on. So we regulate health products. So we have a responsibility for ensuring that different health products are maintaining the standards required to ensure that they are safe and effective. So in terms of, we regulate medical devices, blood, tissues, cells, and organs, and Finbarr touched on that, certain types of controlled drugs. We also ensure, again, that cosmetics maintain kind of high standards. But then we also regulate different types of activities. So we regulate clinical trials. So we ensure that the practice of clinical trials are attaining the high standards and prioritizing patient safety. But then we also regulate the actions of the use of animals in scientific research as well. So again, looking to ensure that the use of animals that the facilities using animals in research are kind of maintaining really high standards in terms of their practice. But for today's talk, I'm really going to focus on the HPRA's role in that process of granting a license to a new medicine. So what is it that we do, and what the companies have to do to demonstrate that they have enough evidence to say we deserve a license and we're going to sell our product on the Irish market. So how does a drug get a license? So it's a very long process. So from that stage, from that early discovery phase that kind of Finbarr was discussing, the identification of a new microRNA, for example, that shows a really promising effect in maybe cancer cells in a lab, to ultimately the generation or the development of a medicine. It could be a pill that ultimately a patient takes at the end of the day to treat their illness or disease. It's a very, very long process. But essentially what the company has to do to get a license, they have to provide three main types of evidence. And the three main types of evidence that companies need to generate over the course of this life cycle, I suppose, from going from your basic discovery to a medicine, the company needs to produce data pertaining to the pharmaceutical quality. So I'll talk about these in a little bit more detail now in a second. So the first package of data that the company needs to produce relates to the pharmaceutical quality. They need to produce evidence in non-clinical models. Again, I'll talk about this in a second. And then the last big package of data that a company needs to produce in order to get a license is our clinical trials, so information derived from clinical trials. And the whole point of these three different packages of evidence that a company needs to produce is they're trying to demonstrate that this new medicine is safe, that it works, and they're able to produce it to a really high standard. So that's really the essence and the core of these different types of evidence that the company needs to produce. And then ultimately, that's then submitted to a regulatory body like the HPRA or the EMA, and then all of that evidence is then assessed. So a bit of a spoiler alert kind of coming up. So the key take-home message from this talk and from the next kind of few minutes is that our job as a health products regulatory authority is to assess all of the data that a company has generated and to actually make sure that the evidence does suggest that the medicine is safe, that it actually works, and it is manufactured to a high standard. So we make sure that any claims that company are making in relation to these kind of elements is actually backed up by data. So that's our primary role in this process. So I'm just going to give you a little bit of an insight into the types of evidence that the companies need to submit to support their application for a license. So the first part of that, I suppose, trio is, can they make or can they produce the medicine to a really high standard? And this comes in their pharmaceutical quality package. So this big chunk of evidence that they have to submit to regulators. And Finbarr, again, nicely kind of set the scene there. So in terms of some companies or some medicines might use cells to produce a medicinal product, or it might be a chemical process that takes many different steps and to produce over time the final product. But during each step in the manufacturing process, so however many steps that takes, byproducts or waste products can be, and very often, produced in that process in terms of the manufacturing of the final medicinal product. So the company has to submit a big package of pharmaceutical quality evidence, and essentially what they have to do is show a really detailed plan and understanding of, first of all, how you produce the medicine. Is it using a cell system? Is it a basic chemical kind of process? So how is the medicine produced? What waste products are produced during this product or process? How do they control the amount of waste products they're producing during the process? So are they implementing processes to reduce the amount of waste products that are produced? How are they removing the waste products? And then again, it's this batch to batch consistency. So it's okay. It's all well and good making one batch of a really pure and potent medicine, but can they do it time and time and time again? So that's a really important part of this type of evidence that the company needs to produce. So is it a pure, is it a potent substance? Can they do it time and time again? And then we also perform inspections to make sure that the data that's actually being submitted to outline all of these processes is actually being done. So we'd have inspectors that go to the manufacturing facility, look at the process, look at all the information, and again, to make sure what they're actually telling and submitting is actually what's happening on the ground. And then finally, a company, because this is a really controlled process, this manufacturing of a drug, it's really controlled. So a company can't just change it or can't change a step. So they're legally obliged to notify a regulatory body, such as the Health Products Regulatory Authority, if they're changing any steps in this process, and they have to provide evidence and data to support that. So that's the first batch of data that a company needs to produce. Can they manufacture this to a really high standard? The next set of data or evidence that a company has to provide comes in the form of non-clinical evidence. So these are really some drugs, depending on the approach, I guess. Some drugs can be really experimental. So back to Finbarr's lab, like kind of this kind of drug discovery of new compounds or new chemical entities. So before we ever test this in a human, it has to go into preclinical assessment. So essentially what we want to do, or the purpose of this preclinical assessment, is to build up a safety profile, to get an understanding of, first and foremost, the impact of the drug on the body. So again, looking down at a cell in a dish, and this compound has great potential to maybe reduce the size of a tumor or whatever the outcome might be. They need to demonstrate this proof of principle in an animal model. So they might take an animal, a cancer model or a mouse model of cancer, give the drug and see whether it has an effect. Another kind of key aspect of this impact of the drug in the body is to understand the safety profile. Does it have an effect on the central nervous system, the respiratory system, or the reproductive system, for example? So again, it's really generating that picture of what impact does the drug have on the body, but then also it's just get an understanding of what impact the body has on the drug. So when you take it, how is it absorbed? Where does it go in the body? How is it broken down? And ultimately, how is it excreted? So animal models or preclinical models are really important for this. Another really important aspect of the preclinical assessment, so again, before we ever test this in humans, is to try and identify what dose do we give. So this is a really nice quote here. So poison is in everything, and in nothing is without poison. So it's the dose that really kind of determines the effect. So again, if we have a mouse model of cancer and we have this really promising kind of drug, if we give it in too low a dose, there's no effect, and ultimately we have an unhappy mouse. On the flip side, if we give too much, we might poison the mouse, and again, we've induced this toxic state. So ideally, we want to identify what's the sweet spot, what is the therapeutic effect where we can maximize the effect of the drug, but minimize the side effects. So the preclinical testing phase is a really important aspect in this development of a drug, and it helps to inform what dose is then ultimately used in humans, which is the final, I suppose, batch of data that the company needs to produce to demonstrate again that they can produce this drug to a really high standard, and then also understand the safety and the effect of the drug. So is it working? So it's the approach to clinical trials is a very progressive approach. So we've gone from that very experimental discovery in the lab. We've tested it in animal models. We have a good idea of the safety profile, but we now start to test this in human models. So give it to individuals to see what, first and foremost, is it safe? What are the side effects? And then ultimately, is it having an effect? So this is a progressive approach. So we have a phase 1 trials, which the primary goal or the focus of a phase 1 trial is really to identify the safety profile, and it's typically given to healthy participants in small numbers, again, because we're just still building up this picture of, is it safe? We then, once we've determined that the safety profile of the drug, well, a good idea of the safety profile in a small number of healthy participants, and we see how the body is responding to the drug, but more importantly, how the drug is responding to the body, we then progress to phase 2 clinical trials, where we're still very much focused on safety, but now we're starting to see, okay, does the drug have an effect? So now we're starting to include patients in the phase 2 trial. So again, relatively small numbers of participants in the trial, but we're checking the safety, but also, is it starting to have a therapeutic effect? And there's also a dose finding element here as well. So we're still at these stages, we're still not sure what the optimal dose is. So there could be a couple of different dosing strategies used here. And then ultimately, again, if the drug, following these 2 phases of kind of investigation, is it safe? Is it starting to have a therapeutic effect? We then expand the clinical trials. And this could be, depending on the drug and depending on the type of disease, this could involve a couple of hundred to thousands of patients. And as we saw in the COVID vaccine trials, there was 40, 50, 60,000 participants in those trials. So really, really big trials. And again, constantly assessing the safety profile of the drug. So safety is paramount all the way through this. But now we're starting to see whether the drug has an effect or an impact on the outcome that we're interested in. So if it's a cancer drug, for example, is it having a therapeutic effect? but also we're investigating it under more real world scenarios where bigger numbers of patients, maybe taking different types of drugs. So really investigating what it's like in a more real world scenario, as opposed to the smaller, more controlled phase 2 clinical trials and then in the phase 1 trials. So once a company has generated all of this evidence, so their pharmaceutical quality data, can they manufacture the drug to a high standard? What is the safety profile in animals? What's the pharmacokinetics? What's the pharmacodynamics? So what's the effect of the drug on the body? What's the effect of the body on the drug? And is it having a therapeutic effect? So once they've generated all of these types of evidence under these three headings, it's then submitted to a regulatory body. And we ask ourselves some very simple questions. Does the data suggest that the drug is safe? Does the drug work and how well? And is it manufactured to a very high standard? And then when we assess the data from all of these different packages, we then ultimately make that kind of question: do the benefits outweigh the risks? Because every medicine has a risk, every medicine has a side effect, but do the benefits of that medicine outweigh any of the potential risks? And that really informs our decision at the end of the day as to whether a company is granted a license to produce. So just to really kind of to drive home the role of the regulatory piece in this, kind of going from your basic drug discovery to the development of a drug that's given to a patient at the end of the day, our role is to assess all of the data, all of the evidence that's generated by the company. And our role is to really make sure that the medicine is safe, does it work, and is it made to a really high standard? And with that, I can see I've just gone over the 15-minute mark. So I'll leave it there, Craig, and hand it back over to you.
So thank you very much, Karl. So our final speaker is Professor Kiran Oboyle. Kiran is in the Royal RCSI, the Royal College of Surgeons in Ireland, and he is the head of the Centre of Positive Psychology and Health. So Kiran, if you want to grab the screen there, we're starting to come through there now.
Good, that should be up with you. Thanks very much, Craig. Let me start my clock here. So good morning, everybody, and thank you for the invitation to be here. So as Craig says, I'm based in the RCSI, and we are, people when they hear about us, they kind of hear the word surgeons, but essentially we're a health sciences university, and we're based in 4 campuses here in Dublin, in Bahrain, and then 2 medical schools in Malaysia. So we take students from 68 countries around the world and train them in medicine, physiotherapy, pharmacy, and so on. And that's all we do. We're exclusively healthcare. And in 2020, we set up a new Centre for Positive Psychology and Health. And what I'm going to do is try to set the context for that. Why would an organization like ours that was set up in 1784, that's a very long tradition in medicine and surgery, why would we now, as a scientific organization and a medical education organization, start moving into this kind of space? And what I'll be emphasizing throughout this is the importance of the scientific foundation of looking at these kinds of areas. Because when you go out onto the internet and you start looking at wellness and well-being and so on, you'll find there's a great deal of material out there that really is not supported by any research. So it becomes important for those of us in the medical schools and the medical universities to start looking at what the evidence is and sharing that with the public. So this talk follows on very well from Finbarr and Karl, in that I wanted to start going out into the community and start looking more at the concept of health. And I think it comes as a surprise to people when they look back to see what the original definition of health is in the WHO establishment documents. And WHO defines health as a state of complete physical, mental, and social well-being. And here's the important part: not merely the absence of disease or infirmity. So most of us tend to think about our health in terms of, well, if we're not sick, we're healthy. But that's not what the definition says. The definition says that there's a lot more to being healthy than just not being sick or not being ill. So you could from that argue that what we have in Ireland is not so much a health service, but we have a disease service, because what happens is people get sick and they go to the service, and the service looks after them, gets them back, hopefully, to not being ill. But there's a great deal more to being healthy than not being ill. And this concept, this way of thinking about this is sort of gathering a lot of momentum now. So if you think about your normal health, and I'm showing that here, here's where the current health service really operates, and the kinds of drugs and therapeutic interventions that Finbarr and Karl are talking about are really focused here. So this is pathogenic. This is really about the origins of disease. So we've signs, symptoms, disability, premature death, and so on. And the whole system is designed to get us to here. But then emerging is huge interest in what we call salutogenesis, and that is the genesis of well-being or health, complete well-being. And there's a whole science now emerging asking the question, how do we move people up to this end of the spectrum? And we'll talk in a moment about lifestyle medicine and positive psychology in that context. So that's the first big kind of idea, that health is much more, it's much broader concept than just the absence of illness or disease. And traditionally treatment has sort of focused down here, whereas this new way of thinking, yes, it's saying we've got to take patients and bring them right, but we've got to bring everybody up in terms of their health and well-being. Now, when we look out there at the incidence and prevalence of disease, particularly what we call the non-communicable diseases, the diseases that we don't get from infections, the situation is really, it's devastating, really, if you think about it. 41 million people each year die from non-communicable diseases, and they account for 71% of all deaths worldwide. And if we take 5 conditions, that's diabetes, cardiovascular diseases, cancer, chronic respiratory lung diseases, and mental health problems, they account for 86% of the deaths and 77% of the disease burden. So it's not just about dying prematurely, it's about living years with a burden of disease. So in the European Union, European region, those 5 conditions account for the majority of deaths and most of the disease burden. The kinds of drugs we've been looking at, that's where the drugs are targeted to help people who have developed these kinds of conditions to manage their symptoms, to improve their quality of life, and so on. But the evidence is that 80% of heart disease, stroke, and type 2 diabetes, and 40% of cancer could be prevented primarily through improvements in diet and lifestyle. So this is as a kind of a refocused way of thinking. The idea here is not so much to treat the condition when it happens, although of course that's terribly important. The idea here is that in addition to that, what we want to do in as much as possible to prevent the condition developing in the first place. And central to that is the individual. Central to that is each and every one of us. It's the idea of empowering us to manage our lifestyles in such a way that we don't develop these kinds of chronic conditions. And in medicine and nursing and so on up to now, that's really been the Cinderella side of intervention, prevention. But it's becoming more acute because we can't keep going the way we are, because as countries develop economically, the amount of non-communicable diseases increases as lifestyle changes, and also as populations age, we're seeing far greater numbers of people becoming chronically ill. So we're not going to be able to continue to cope with the kind of healthcare system that we have now. And we've seen even with COVID how, you know, not a lot is required to put huge pressure on our healthcare systems. Now, if we look at the picture in Ireland, this is from the HSE's Healthy Ireland report published about 2 weeks ago. You'll see that over 1 million of us currently have one or more chronic diseases. And that's a huge number. 49% of us have one chronic disease and 18% have more than one. And our rate of development of chronic diseases is increasing at about 4% per year. The most common diseases in Ireland are cancer, cardiovascular diseases, heart and blood vessel diseases, and respiratory or lung diseases. So it's a very, very sobering picture when you look at it in terms of these kinds of numbers. And a very nice report from McKinsey just last year, looking at the economic consequences of chronic diseases for countries, basically estimated that the payoff, the economic payoff for a country, never mind the humanitarian and the welfare payoff for individual patients, the economic payoff of prevention is absolutely huge in real terms, in real economic value. And to do that, this is why I think these kinds of seminars are very important. We've got to make health a social and economic priority. We've got to keep health on everyone's agenda. We really do need to transform our healthcare systems. And we need to double down on innovation and therapeutics. So it's not one or the other. It's really both. It's the kinds of things that we've heard about already this morning, plus a change in the way we think about prevention. And this kind of thinking has led to the emergence of two kinds of key specialties. One is lifestyle medicine, which is the application of environmental, behavioral, medical, and motivational principles, getting people to change their behaviors, to the management of lifestyle-related health problems in a clinical setting, including self-care and management. So we're now seeing emerging this specialty called lifestyle medicine. And it's very interesting that the big health insurance companies are starting to adopt this, because from their business model point of view, it makes sense. If you can prevent people getting sick for longer, your costs go down, cost of maintenance and interventions, and so on. And here's a very nice quote from a paper by Katz back in 2013. Now, he said, an aggregation of evidence, basically from modern epidemiology, says that tobacco, poor diet, and lack of physical activity constitute the leading causes of chronic diseases. And then Katz goes on to say, and I think this is really nice, he says, conversely, the salutary use of feet, fork, and fingers. So that's something to remember. Feet for physical activity, fork for diet, and fingers for not smoking, not overusing alcohol and other drugs, could slash the risk of all chronic disease by 80%. Those kinds of lifestyle interventions. And it's more complex even than that, because we know that the genes that we're born with that contribute to these conditions, the expression of those genes, whether they become active or not, is also influenced by our lifestyles, either for negatively or positively. So it's possible to influence dangerous genes, if you like, by changing our lifestyles. Okay, so then we ask the question, well, what's psychology got to do with it? Because that's the second big arm of this. Well, the first is we're becoming much more aware of the complicated relationships. Brains and minds and bodies are not separate entities. They all work as one. We know non-communicable diseases are growing, but the risks are behaviors. So diet, lack of exercise, sleep, drug use, alcohol use, they're all behaviors. And psychology is the science of behavior. And then in addition, we know that mental health is emerging as a major challenge in the developed and developing world. So we've got to start figuring out, how do we improve the mental health of everybody, not just people with mental illness. So positive psychology then is a scientific discipline, and really important to emphasize that looks at the upside of the human condition, happiness and well-being. So we use the scientific method to explore and enhance the positive aspects of life. So it's the same kind of ideas we were talking about earlier with physical health, that the absence of mental illness, which is what we tend to treat here traditionally, is not the same as the presence of happiness and well-being. So yes, we're interested in positive psychology in people with mental illness, but we're interested in improving the lot of everybody. And if you ask the question, well, can that be done? What do you inherit your level of happiness? The evidence is you do. About 50% of your level of happiness is inherited, but you can influence that to about 40%. By 10% from your circumstances, and then 40% by how you live your life. Now, I don't have time to go through all of this, but you'll have it on the slides. When we talk about happiness, we're not just talking about feeling good, positive emotion. While that's important, engagement, doing activities that are interesting and absorbing for us is key. Relationships are key. having a sense of meaning in what we do is key, and then having a sense of accomplishment, of achievement in what we do. And the evidence now from positive psychology, I just want to give you examples of these here, is that we can intervene ourselves as individuals in ways that increase our level of happiness across each of these 5 areas of happiness. So, for example, relationships, improving relationships to improve compassion and communication skills, meaning considering your values and goals and purposes, achieving true self-discipline, and so on. So really what I'm saying here is we have scientific evidence to show that interventions of this type actually significantly increase happiness and well-being. And if you want to follow up on this personally, the best website I think you can look at is the VIA Institute of Character in Cincinnati. Really good science behind this. It's an NGO. We know the researchers there. You can log on there and do a detailed questionnaire which lets you identify your strengths. And we tend very much to focus on our vulnerabilities and on our weaknesses. The evidence is that by finding out what your strengths are and focusing on those, that can significantly ramp up your sense of well-being. So there's an example of one well-validated, strong scientific evidence for the intervention that you can access immediately. Oh, and by the way, it's free. And for any of you who have children, there's a version of the scale for 10 to 17-year-olds, which really works very well, helping kids to identify their strengths. And this is important because not only are strengths and, you know, kind of things like optimism related to our mental health, they're also related to our physical health. There's a huge study published last year in the Journal of the American Medical Association showing a link between optimism and cardiovascular disease and all-cause mortality. And in a nutshell, basically the evidence is that optimistic people don't develop cardiovascular disease to the same extent as pessimists, and they live longer. Why is that important? We can teach people to be optimistic. Even pessimists can learn to change their mindsets to become more optimistic. Final 2 points I want to make here is when we start to look at the linkages between our brains and our bodies, one of the linkages we're very interested in is something called metaformation. And that is this low-grade type of inflammation that was discovered in the mid-90s. That's quite different from this kind of inflammation you get here when you get a virus or a bacterium. This is a type of inflammation that goes along at a low level but causes damage. And it's triggered by stress, it's triggered by diet, it's triggered by lack of physical activity. So we're starting to learn now what are the mechanisms that translate these kinds of lifestyle factors into disease. There are fascinating research, major center in Cork, Ted Dinan and John Prine, is linking what happens in our gut to what's happening in our brain. And there's some fascinating research emerging there about how our guts produce the kinds of chemicals that our brain needs to function normally. So that's all I'm going to say about that. Anybody who's interested in following up further on this, if you go to the RCSI website, we've a Science of Health and Happiness program there that's free. It's 10 lectures. 35,000 people have registered for that as we speak. We have a Science of Health and Happiness program for young people. And then we also have the RCSI My Health series, where we explore a lot of these relationships. My final takeaway is that this is really about changing the way we see things, I think. Marcel Proust said that the only real voyage of discovery consists not in seeking new landscapes, but in having new eyes. So we're not saying that we move away from therapeutics and drug development, biotechnological development. We're saying that in addition to those, we start to really focus on preventive health and helping people to improve their lifestyles. So thanks very much. I'll stop sharing there, Craig.